Thank you, Dr. Miyashita, for your introductions, and I'm very happy to take this opportunity as a final symposium on Tohoku Forum for Creativity. So I'm going to talk about mammary gland biology today. So the defense system in mucosal tissues, such as gastrointestinal tract, respiratory tract, or vaginal tract, would be very much important for protect our bodies from infectious diseases occurring in mucosal tissues, such as influenza, or diarrhea, or HIV, and so on. That's why it's well known that such mucosal tissues possess well-developed immune system. The recent advance of metagenomic approaches using a new generation sequencers allow us to identify microflora in the mucosal tissues. So now we believe that both immunological and microbiological environment would be crucial for the protection from infectious diseases. So in the mammary gland that I would like to talk to you today in this presentation is one of mucosal tissues. However, compared with other mucosal tissues, the developmental process of mammary gland immunological and microbiological environment have poorly understood. That's why it's very important to think about how we protect the mammary gland from infectious diseases. In order to tell you why we are very interested in studying the mammary gland biologies, I need to use 1 or 2 minutes in order to tell you the dairy farming system. To do so, I want to ask you to be dairy farmers. So you need to do first is to do the artificial inseminations in order to get milk. And pregnant period of dairy cattle is usually 28 days. That means after almost 9 months, you can get baby from your dairy cattle. And then you can start collecting milk. And y-axis of this graph shows you how much milk can be collected in a day. And usually, the early stage of dry lactation period, amount is not so high, but it's usually gradually going up and then reach to the maximal level, and then going down. And you usually collect milk for 10 months, that is lactation period, and then we stop collecting milk. That is drying period. And in order to start second cycle lactation period, you have to do the artificial insemination during the first lactation period, and then you can start collecting milk. That is second cycle lactation milk. And in usual, we can get 8,000 kilogram milk during one lactation period. So let me calculate how much money you can get from 8,000 kilogram. So you usually sell 1 kilogram, 1 liter of milk, like $0.8. That means you can get $6,400 from one lactation period for 10 months. But of course, you can't get all because you need to pay for diet, or you need to pay for animal facility maintenance, or if you have people who can help you, you need to pay for them. So maybe the amount you can get is not so high. And another thing that you have to consider is that if you want to have new pregnant dairy cattle, it's very expensive. You have to pay a lot. That's why most dairy farmers want to repeat the lactation period several times, like 5, 6, 7. But point here is that the average of lactation period in Japan is not so high. That is about 2.7 because of infectious diseases occurring in the mammary gland. That is mastitis caused by infection with, for example, Staphylococcus aureus or E. coli. And the economic loss of mastitis is so huge in Japan, or in the world, that is 7 billion in Japan. That's why it's very important to control the mastitis. Well, it's very important to understand how we can protect the mammary gland from infectious diseases, infections. So mammary gland is one of exocrine organs, but compared with other exocrine tissues, the development process is very unique because the mammary gland development process is only initiated after sexual maturations. And after sexual maturations, mammary gland get like sex hormone-derived signals. For example, estrogens. Estrogens. And estrogen facilitate to develop the mammary gland duct and adipose tissues. But at this time point, this mammary gland still doesn't function. And after pregnancies, the mammary gland gets another signal, sex hormone signals, that is progesterone. And this hormone can facilitate the development of mammary gland alveoli, like this. But in this stage, it still doesn't function. But after parturitions, they can get more signals, such as prolactin, growth hormone, insulin, and glucocorticoid, whatever, moreover. And then the mammary gland alveoli tissue is more maturated, and then we can start to produce milk proteins. That is functionally maturated stage. And then once animal is weaning, the mammary gland structure is immediately egressed, like this. So now you learn that how mammary gland develops. But if you do the histological analysis, you can more precisely appreciate the maturation process of functional development or morphological development. And this slide shows you the mammary gland structures when you take the tissues from non-pregnant mice before sexual maturations. So you can only see the mammary gland duct around here, and most tissues are adipose tissues. But once the animal is pregnant, you can clearly see the maturation tissue, mature tissues, and then you can see the structures. But in this time point, during the pregnancies, it still doesn't function, as I told you. But after parturitions, the mammary gland alveoli structure is more matured, and then become bigger, and then they start to produce milk. And then after weaning, the structure is immediately gone, and then they stop to produce milk. And this, I think that in addition to the uniqueness of developmental process of morphological maturations, you can also learn functional maturations, because most important function that mammary gland has is to produce milk proteins, such as caseins, from mom to the babies. And other important thing that you have to think about is to transfer antibodies from mom to the babies. So if you are interested in looking at the functional maturations in the mammary gland, you can do the quantitative PCR analysis in order to investigate expression level of alpha or beta and kappa casein expressions. So specifically, we collected mRNA from the mammary gland that obtained from different stages of mammary gland, from non-pregnant, during lactation, and after weaning, and then performed the qPCR. And as you expected, if you look at the casein expression before pregnancy, during pregnancy, after weaning, at that time the mammary gland doesn't function. You don't detect any expressions of caseins. However, if you look at the casein expression during lactation, you can clearly see the expressions of all three caseins. And the expression level is gradually going up and then reaches to the maximum level like this. That means that the mammary gland only functions during lactation, and then functional process or maturation process of mammary gland functions is gradually accelerated during the lactation period. So now, I think some of you notice that you agree that actually I'm true, because my most important function that mammary gland has is to produce milk protein. But I think some of you notice that, well, actually it's true, but only thing that mammary gland cannot produce is antibodies, because antibodies should produce B cells, or I should say plasma cells. That's why if you produce antibodies in the mammary gland needs to get B cells or plasma cells in order to produce antibodies to transfer from mom to the babies. So to address these questions, we performed next experiment. But to explain this experiment, we have to tell you that the most important immunoglobulin that is produced in the mucosal tissues is IgA, because IgA level is much higher than IgM or IgG. So it's quite important to look at the expression level of IgA in the case of mucosal immune system. So our next experiment to look at how much IgA producing cells are present in this mammary gland throughout the reproductive cycles. So this is immunohistochemical analysis. So if you see the brown cells, that means you have IgA producing cells in the mammary gland. And this slide shows you the mammary gland tissues when you get the tissues before pregnancy, after sexual maturations. But I think you don't see any brown cells. That means that's true. That means you don't have any IgA-producing plasma cells in the mammary gland tissues before pregnancies. But if you take the cells during the pregnancies, actually you can see the structure that is mammary gland alveoli immature, but you still don't see the IgA-producing cells shown as brown colors. But if you take the tissues during the lactation period, so you can first see the brown cells, that is IgA-producing cells. And the number of IgA-producing cells is higher when you study 2 weeks after parturition, but you don't see any IgA-producing cells when you analyze just after parturition. And then once weaning, you lose IgA-producing cells. That means that mammary gland can recruit the IgA-producing plasma cells in order to produce the antibodies to transfer from mom to the baby. And this is quantitative data, so we counted and how much numbers of IgA-producing cells are present in the mammary gland tissues. And as you know already, so the number of IgA-producing cells is much, much higher 2 weeks after parturition, but other stages, it's almost zero. And to study the B-cell maturation in the mammary gland, you can also do the flow cytometric analysis, because if you use 2 types of B-cell markers, such as IgA or B220, you can more clearly understand the B-cell maturation process. Because most plasma cells, shown here, are IgA positive but B220 negative. But these plasma cells are differentiated from plasmablasts, and then plasmablasts are differentiated from mature B cells that are basically present in the secondary lymphoid tissues. So when you do the flow cytometric analysis using these 2 types of markers, you can distinguish mature B cells, that is naive B cells, or plasmablasts or plasma cells. So what we did was to isolate the cells from each stage of the mammary gland and then stain the cells with anti-IgA or anti-B220 in order to look at how much frequency of these 3 populations are present in the mammary gland. And what we learned was when we take the cells from before pregnancies, like this, in this stage, we don't have any B cells or plasma cells. And during the pregnancy, we still don't have any B cell plasma cells. But just after parturition, the number is a little bit increased, and then a little bit more increased. And 2 weeks after parturition, we have very high level of plasma cells. And then once weaning, we lose the plasma cells. But when we look at the mature B cells, or plasmablasts around here, the number is very, very low. And this is absolute numbers. You can clearly see the abundant number of IgA-producing plasma cells 2 weeks after parturition. So these results clearly indicated that actually we have IgA-producing plasma cells in the mammary gland during the lactation, particularly 2 weeks after parturition. But these kind of cells are somehow recruited into the mammary gland from somewhere in order to deliver the antibody from mom to the baby. So we next wanted to know if we have IgA-producing plasma cells in the mammary gland, and then these populations are essential to produce antibodies, other maternal antibodies, we should get very high level of IgA in the milk. To address these questions, we performed ELISA analysis using milk collected from different stages of the mammary gland. So in this case, we collected milk 0 weeks just after parturition, 1 week after parturition, and 2 weeks after parturition, and then performed the quantitative ELISA analysis in order to look at the concentration of 3 different types of immunoglobulin. And what we learned was actually consistent with flow cytometric or immunohistochemical analysis. The amount of IgA 2 weeks after parturition is very high compared with other stages. And actually we can see some quantitative difference, but amount of IgG or IgM is very low compared with IgA, suggesting that IgA is the most important immunoglobulin in mice during the lactations. So next question we had was how the B cells or how IgA-producing plasma cells will actually recognize the mammary gland situation. I mean, when now the mammary gland is lactation period, and then they start to move into the mammary gland. To address these questions, we designed a simple experiment using two models. So one is normal lactation models, and the other is forced weaning models. So in this case, we had pregnant mice and then get the babies, and then maintained mom and pups together for 1 week. And then 1 week after parturitions, we take the pups out, and then just mom was maintained for one week. And then we collected mammary gland tissues at this time point. And what we did first was to see the histological change of the mammary gland structures. And what we learned was that actually, as I mentioned, we have mammary gland tissues 2 weeks after parturitions. You can clearly see the developed mammary gland structures. That is much more development compared with just after parturition that we use as a control. But most surprisingly, when we look at the morphological structures of mammary gland in this forced weaning group, the structure was immediately disrupted. That means that suckling signal is very much important to maintain the mammary structures. And our next question is, whether or not the B cells or IgA-producing plasma cells are actually present in this forced weaning group. And then we did immunohistochemical analysis again, and then found that actually IgA-producing plasma cells are very low in this time point compared with this normal lactation group. So these results indicated that suckling signal is very much important for the recruitment and maintenance of IgA-producing plasma cells in the mammary gland. And this is a quantitative and counting numbers, so you can see dramatic difference between the normal lactation and forced weaning mouse group. So next questions we had was how IgA-producing plasma cells actually can recognize the mammary gland-derived signals in order to infiltrate into the mammary gland tissues. To address this question, we have to know the chemotactic activities produced or secreted by mammary gland. Because it's known that actually mammary gland produce one of chemokines named CCL28. And then the B cells or plasma cells can recognize this CCL28 derived from mammary gland. And then once recognitions, they start to migrate into the mammary gland. So we hypothesize that actually if our data is true, maybe the expression level of CCL28 is going up during lactation. That's why we have so many IgA-producing plasma cells in the mammary gland. So to address this question, we did an experiment. So in this case, we collected mRNA from normal lactation groups before pregnancy, during lactation, and after weaning. And we also collected mRNA in the forced weaning group 2 weeks after parturitions. So let me remind you how much amount of IgA-producing plasma cells are present. So I just want to show you the plus or minus. So again, plus means high level, and minus is almost 0. So our hypothesis was maybe if we analyze CCL28 expressions, the data profiling pattern is almost similar to the number of IgA-producing plasma cells. And actually this is true because when we did the qPCR analysis in order to look at the expression level of CCL28 in the mammary gland, actually we have very high level of CCL28 expression during lactation, particularly 2 weeks after parturition around here. And the level is going up, and then after weaning, forced weaning, normal weaning, the level expression level is dramatically or immediately going down. So meaning that CCL28 would be essential for the recruitment of IgA-producing plasma cells into the mammary gland. So our next question is, what receptor is involved in the recognition of CCL28-mediated chemotaxis? And we searched some literatures and found that in humans, there are 2 receptors that can react to the CCL28. But it's still unknown that in mice or in the mammary gland, which receptor is involved in the recognitions. So we took advantage of using the knockout mouse models. So in this case, we use wild-type mice and CCL10 knockout mice or CCR3 knockout mice or double knockout mice, which lack both genes. And then we collected milk in order to evaluate how much milk IgAs are present or produced. So again, we have lots of IgA in milk when we collected milk from wild-type mice. And also, if you take the milk from CCR3 knockout mice, you have equivalent level of IgA. However, most surprisingly, when you take milk from CCR10 knockout mice or CCR3 or CCR10 double knockout mice, the IgA level is almost non-detectable. That means that CCR10 knockout mice, IgA production is almost abolished. So these results clearly indicated that CCL28 and CCR10 axis would be crucial for the recruitment of IgA-producing plasma cells into the mammary gland. So now I would like to change a little bit my gears in order to talk about the microflora in the mammary gland. Because as I mentioned, so in the mucosal tissues, we believe that both immunological and microbiological environment would be very much important to protect from infectious diseases or infections caused by pathogenic bacteria. So we hypothesized that mammary gland should have microflora, and this kind of microflora is very much important for not only protection of mammary gland, but also deliver the mammary gland. I'm sorry, not for protection from mammary gland, but also deliver the mammary microflora from mom to the babies. And then that facilitates the development of microflora structures in the gastrointestinal tract in the infant. So to address these questions, that is, the actually mammary gland possess the microflora during lactation. We did the next experiment. So in this case, we collected genomic DNA from different stages of mammary gland, from pregnancy, before pregnancy, during the pregnancy, lactation period, and after weaning. That is almost same to the previous experiment. The only difference is, in this case, we collected genomic DNA, not mRNA, and then performed the qPCR again in order to look at TUF gene copy numbers. Because TUF gene is identified as a bacterial-specific gene that is only localized on the genome, just one copies. So if you see the one copies, that means you have one bacteria in the tissues. And this is a result. So if you look at the level or copy numbers of TUF genes in the mammary gland throughout the lactation period, you can clearly see the high numbers of TUF gene during the lactation. And you still have some level, but it's a little bit different during the lactation compared with other stages. And then we wanted to know what kind of bacteria are present in the mammary gland, because we have bacteria. Then we did a metagenomic analysis using new generation sequencers and identified that if you take the DNA just after partitions, or if you take the DNA 2 weeks after partition and then perform this kind of analysis, you have very developed microflora in the mammary gland, which consists a large number of large species of microorganisms. And mostly Bacteroides, Clostridium, and Lactobacillus are present, all of which are well-known intestinal microflora. So suggesting that actually mammary gland have immune system, and also mammary gland have microflora during lactations. And then finally perform the very simple experiments in order to study the influence of microflora on the development of mammary gland immune system. So in this case, we treated mice with antibiotics in order to remove the gut microorganisms and then look at the expression level of IgA or number of IgA-producing plasma cells in the mammary gland. And this is one of the data that we have. So if you take the cells and stain with IgA and B220, that is a marker for B-cell maturations, you found that you have lots of plasma cells, IgA-producing plasma cells in the mammary gland. Of course, in the wild-type normal group, just treated with distilled water. However, if you take the cells of mammary gland of mice that was treated with antibiotics, the number of IgA-producing plasma cells is dramatically decreased. That is a significant difference. And then consistent with the number of IgA producing plasma cells in the mammary gland, when you take the milk and then perform the ELISA analysis in order to look at the amount of IgA in milk, you can also see the difference between two groups, suggesting that the intestinal microflora might contribute to IgA productions in the mammary gland. So take-home message. Actually, because of time limitation, I couldn't show the all data, but I also don't know the whole mechanism of how mammary gland immune and microbial environment develop. But I can only say that mammary gland has super special or unique program that facilitate to the development of immunological and microbiological environment during the lactation. And then understanding the development process of mammary gland immune and microbial environment would be very much important for the improvement of quality of breastfeeding, and also important from protection from mammary gland tissues from infectious diseases. So finally, I would like to appreciate my students because they did lots of experiments, hard work. Especially, I would like to super thank Kanae, Katsuki, Yuki, Miku, Mutsumi, Shiho, Shun, Yuri. They did wonderful studies. And also, I would like to thank CFAI faculty members because this kind of studies was done as a collaborative studies in CFAI. Especially, I would like to thank Dr. Kitazawa, Yoneyama, Suyama, Sakai, Watanabe, Dr. Aso for their kind contributions. And I also appreciated my previous mentor, Dr. Kiyono, and his colleagues because he gave me lots of chance using knockout mouse model. And then finally, I would like to appreciate JSPS for their funding support. Thank you very much for your kind attention. I'm very happy to take your comments.